If everyone would come in and have a seat. Welcome to Seminars in Oncology. It's my pleasure to introduce our speaker this afternoon, Dr. Andrew Ewald. I have known Andy for a long time. We were good friends and colleagues back at Johns Hopkins, which is where he's really spent his faculty career. He did his undergraduate at Haverford College, majoring in physics and biophysics as a minor. I had forgotten your background in physics. And then went to Caltech for his PhD in biochemistry and molecular biophysics, then did a postdoc at UCSF, and that's where he really found his love for epithelial biology, started researching in breast cancer. He's now an incredibly accomplished investigator in that field. I'm really looking forward to his talk this afternoon. It'll touch on, as you see in his title, cell states and tumor microenvironments and their control over breast cancer metastasis. He is at Johns Hopkins the founding co-director of the Cancer Invasion and Metastasis Research Program at the Cancer Center, as well as the director or department chair for the Department of Cell Biology, now 15 faculty strong at the Hopkins Medical School. I always will remember Andy, even as a very young faculty member, being one of the most thoughtful people about ways to create intersections and communications between basic scientists at the School of Medicine and investigators at the Cancer Center and physicians and clinicians doing research. He has the title now of the Virginia Diacetus Professor of Basic Cancer Research at Johns Hopkins. He previously had a number of important awards from the Institute and from societies, including the Johns Hopkins University Daniel Nathan Scientific Innovator Award. He's been an invited speaker for the Nobel Foundation Congress on Cancer Metastasis in Stockholm, Sweden, given the Blaffer Lecture at MD Anderson. We're just really delighted that you would spend the day with us at the Farber and look forward to your talk, Andy. Welcome.
Thank you, Kathy, for the very generous introduction. You're still remembered very fondly at Hopkins, and my thoughtful is actually the first word comes to mind from about you for me as well. And we are still figuring out how to implement, quite literally, all the suggestions she made for the physician-scientist tracks at Johns Hopkins. It's a pleasure to be here today. I'm going to talk to you about metastasis. As everyone here knows, metastasis is the process by which cancer spreads in the body. If there's one thing you take away from this seminar, I want it to be that metastasis is not a single step. It's not just about getting faster or migrating more efficiently or grabbing onto the ECM. It's more like the decathlon in the Summer Olympics, where you've got 10 distinct events you need to do. There's different equipment you need, there's different skills you need, and the best metastasizing cell is the one that can change its behavior in order to accomplish the different steps of growth, invasion, escape from the tumor, entry into the circulation, survival. We don't think about this often enough. An epithelial cell lives connected to its neighbors. It doesn't live in a solution environment, right, in any normal context. And so it's got to survive in a liquid environment, get out, evade the immune system, and grow to significant size. For purposes of today's lectures, I want to really challenge you to think that there's a lot of constraints that the nature of epithelial architecture places on epithelial cell behavior, right? It's easy to say, oh, the cancer cell metastasizes, and to forget that those cells are attached to each other by multiple classes of intercellular junctions, that in the case of the mammary epithelium, there's additionally this myoepithelial layer and then a basement membrane. And we're going to think about different aspects of those constraints and how the cancer cell manages to escape those constraints to travel through the body anyway. So how do we approach this problem? Our lab is very interested in the use of 3D culture. We have been for the last 20 years, interested in the use of 3D culture to make events that would normally be unobservable and unmanipulable in the body into something that we can study every day, watch in real time, and intervene through a variety of molecular and experimental techniques. So the core approach we're using across most of our assays, whether it's a mouse tumor, a human tumor, breast tumor, pancreatic tumor, the core approach we're using is to isolate a fresh primary tumor, chop it up with a scalpel, digest it with enzymes, and get pieces of mammary tumor organoids, right? So these are 200 to 300 cells. The word organoid is used a lot, and it's used to mean, unfortunately, it's used to mean very different things. So for purposes of today, Ewald lab organoids, these are acute cultures. We're not passaging, we're not freezing, we're not biobanking. We're trying to study the in vivo state rather than create an indefinite resource. Claver-style organoids are much better for creating indefinite resources that are biobankable and shareable. Because we're just trying to study it for a week or two, we can use very simple media conditions that don't require Wnt, don't require us to push the phenotype of the cells in any particular direction. We also get a sizable starting material on day one. From a clinical sample, we can get 500 to 5,000 organoids. From a PDX tumor, 25,000. From a genetically engineered mouse model or a GEM, 250,000. So that lets us set up many different experimental conditions with tissue randomly allocated from the same tumor. This is working in mouse and human, primary and metastatic site. We started in breast cancer. It's been adapted to many cancer sites. And the only purpose of the list of papers that are methods-oriented at the bottom is to say that our methods are very open. We've published exactly how we do this. If it's unclear, please contact us. All right, let's jump in. So this here is a piece of a stage 3C breast tumor. Right? Patient was diagnosed stage 3C breast cancer. That's about as aggressive as you can possibly get without already being frankly metastatic. This patient had 20 lymph nodes sampled. All 20 had cancer cells growing in them. And so what's, I think to me still years later, quite surprising, is how normal this growth looks. Right? You have a piece of a highly disseminated tumor that's sitting there and it's growing, almost doing a program of branching morphogenesis. So that led us to ask this question of whether the ECM microenvironment around the tumor could be an important regulator of whether it invades or not. And I will say, that wasn't how I came to the problem. I assumed that if you got a highly invasive tumor, it would invade in a variety of contexts. I was surprised by the previous slide. So we tested this. We tested lots of different 3D environments, but the two that I'm going to compare today are the basement membrane-like gel, so 3D matrix gel, collagen 4, laminin 1, these sorts of things, like what would normally surround an in situ tumor, versus stromal ECM, which is what the cancer cell is going to see as soon as it gets to the other side of the basement membrane. These are pieces from the same human tumor. The media is the same. They're imaged at the same time. So if you see a difference on the next slide in cell behavior, it's to do with the structure and organization of the extracellular matrix. I realize not everyone at the Farber is a pathologist, but which one of these looks worse? You know, it's the one on the right. I think in a sense, if you watch the timer as it goes around this time, it's one of the most terrifying things my lab's ever done. Simple, very, very simple experiment, but this is fast. Wrong genotype plus wrong microenvironment, it's really fast. The thing explodes. The image on the left, remember, those cells are just as bad, right? They were randomly allocated. If you put them in a mouse, it's very likely to form a tumor, right? And you put it in this invasion-suppressing microenvironment, they sit still, they form a nice epithelial edge. So I hope that it presses upon you, though it's a very simple experiment, that the context around the cancer cell can be a very important regulator of its phenotype. So what do we learn there? That the invasion is a tissue-level phenomenon that emerges from dialogue between the cancer cell and the ECM. Invasion requires, as we'll get to in the next section, molecular potential in the cancer cell. Not every cancer cell is capable of invading, even if you put it in a collagen gel. And it also needs permissive signals from the microenvironment. The basement membrane contains molecular signals that can suppress invasion, and even highly aggressive cancer cells, right? Stage 3C breast tumor, right? You put it in that matrigel environment, it's still listening, it's able to respond to and respect those constraints. So we had a whole line of experiments, they're still ongoing, looking at the role of the tumor microenvironment on the cancer cell. We've also flipped the logic where we say, okay, we know that if we have a highly fibrillar collagen 1 microenvironment, it's going to promote invasion, right? So we flipped the logic in the next set of experiments, and what we're trying to do is put cancer cells randomly allocated from the tumor into that invasion-promoting environment and force the cancer cells to teach us which ones are most invasive and what are the molecular programmings they're utilizing. The analogy I want you to think of is the Boston Marathon. If you go to the elite runners division, right, they're all fast. They're all wearing the right running shoes. They all want to win. Predicting's really hard. That's what you get when you look at an H&E section. You have to sort of guess which of these cells is most invasive. Here, you don't need to guess. You know, just as in the Boston Marathon, if you look at mile 26.1, you can predict the winner really easily. It's the guy in front, and a tenth of a mile later, he wins. Here, you can look at the cell that's leading the invasive column, and hypothesized pretty accurately that it's more invasive than the cell in the bulk. We get 250,000 of these per gem tumor. They're single transgene models, so we have racks of them. So you can literally test this millions of times and just see what holds true on the average. So the mammary epithelium is composed broadly of two compartments, an inner luminal compartment, this forms the ducts, and then eventually the alveoli that make the milk. There's also an outer basal compartment that consists of stem cells and myoepithelial cells. The myoepithelial cells contract to expel the milk. Medical oncologists also think about breast cancer and treat breast cancer in terms of this luminal versus basal dichotomy. And so it's very reasonable to think, what's going on in a luminal model? So we have a luminal mouse model. We look with a luminal gene like keratin 8, right? And we see that it's there. It's not surprising. It's effectively a tautology. It was named that because it had high expression of these genes. It was very surprising that leader cells also expressed basal genes like keratin 5, 14, p-cadherin, and p63. I want to highlight these cells do not express smooth muscle genes. They're not myoepithelial cells. It's a luminobasal leader phenotype. The keratin 14 negative luminal cancer cells can induce keratin 14 positive expression. So we're seeing state changes between keratin 14 negative and positive. And it's those newly keratin 14 positive cells that lead collective invasion. These cells are present in the luminal model in vivo. So you look, okay, mostly blue, luminal, some green, right? So you have some green, but it's only about 1.5% of the cells in the tumor. But where they're doing bad things, that's where we're seeing the basal phenotype. We knock it down, the invasion is blocked in vitro, invasion is blocked in vivo, right? So you see there's the keratin 14 positive invasive cancer cells at the edge versus a smooth epithelial edge. Interestingly, it also resulted in a large reduction in expression of broadly pro-metastatic genes. So you end up having here, the background I wanted to set you up with was that you can have these states of epithelial differentiation doing different things at different stages. Keratin 14 negative state bulking up the primary, keratin 14 positive cells spreading through the body, and then a keratin 14 negative state bulking up in the distant site, right? And I want to highlight that an single intermediate filament protein, keratin 14, was actually regulating the pro-metastatic cell state. You take it away and there's broad transcriptional changes. How does this relate to distant metastasis, right? How does this relate to the ability to spread through the body? Well, these invasive strands, which can feel like they're just a static arm at the edge of the tumor, right? They only look that way if you kill them first. If you look at them in real time, you actually see they're highly dynamic. They can launch individual cancer cells. And if you create primary tumors with a mixture of colors, what you can see is that the collective invasion strands have mixtures of colors, at least trivially multiclonal, that launch groups of cancer cells into the stroma, into the blood, and form multicolored micro and macro metastases. So when we take that together, we have metastasis arising from groups of cancer cells that initiate at least diverse, potentially multiclonal, it depends on your definition of a clone, metastatic tumors. So you're seeing collective invasion and dissemination of clusters, expression of a requirement for epithelial genes, at least keratin 14, and that the metastases can initiate with genotypic and phenotypic diversity, which could complicate treatment response. So that's a quick summary of a series of work that we did to try to define how things worked in luminal breast cancer. We were able to show that this held true across the subtypes, that was observable as a mechanism across the subtypes of breast cancer, both in mouse models and in patient tissue. But we anticipated that it was unlikely that it was going to hold true in all contexts, that it would explain all of breast cancer or all of cancer. So we wanted to figure out what would be some interesting contexts that we could look at where we might hit the limits of this keratin 14 model, right? Where we might see different biology. At this part of the talk, I'll talk about triple negative. At the end, I'll get to Claudin low. So we thought triple negative was an interesting place to look. First of all, it's important, right? There's a great clinical need because this, by definition, lacks estrogen receptor, progesterone receptor, and HER2. Those are the targets for most of the therapies used in breast cancer. The lack of those receptors greatly limits what's available for treatment. It's also associated with rapid metastatic recurrence. And then it was fair to think that it could be a setting where we saw exactly the same thing, right? Because most triple negative tumors display basal gene expression. It was also possible we would see something very different because it was clear that EMT markers, if you looked across the subtypes of breast cancer, EMT markers were frequently detected in triple negative. So the challenge with epithelial and mesenchymal markers is, first of all, which ones to look at. We looked at many. I can tell the story simply with two, so all the slides will focus on E-cadherin as a model for epithelial, and Vimentin as a stand-in for mesenchymal. But one of the things that's challenging is to try to understand which is the cancer cell and which is the stromal cell, and when are you seeing co-expression in the same cell versus expression in adjacent cells. So in the luminal models, and these are genetically engineered, we know the oncogene, we're therefore able to use antibodies to detect the cancer cells, so we can be unambiguous in our assignment of cancer cell identity. We look and we say, okay, we're seeing epithelial phenotype cancer cells, we're seeing Vimentin-positive stromal cells that are adjacent. We look at triple-negative breast cancer, we see something very different, where we're seeing double positive at the single cell level. We're seeing epithelial cancer cells where they're expressing both E-cadherin and Vimentin. So what we wanted to do at that point was do that same Boston Marathon, put them in a pro-invasive environment, and just see which cells lead. Because if you look at a different core of a triple-negative breast tumor, it might be much less mesenchymal. What we saw when we looked at Vimentin as a stand-in for mesenchymal identity was in the luminal models we looked at, it had no relationship to invasive behavior, right? There's occasional Vimentin-positive cells. They're rare if you deplete the stroma, right? The epithelial cancer cells with Vimentin are rare in luminal, and they don't relate to the invasive pattern. We saw something really different in the triple-negative breast tumors, right? So in the genetically engineered mouse model or either of two PDXs, which is that the cells leading collective invasion had an E-cadherin Vimentin double positive hybrid EM phenotype. Consistently, GEMs, PDXs, fresh patient tumor tissue. So we wanted to then test whether EMT was required for triple-negative breast cancer invasion. But I couldn't figure out how to do that. I still don't know because EMT is a broad concept. There's many relevant transcription factors. So we decided to ask a narrower question, which was, is Vimentin required for triple-negative breast cancer invasion? This was still interesting to do because there was significant literature suggesting that Vimentin-positive cancer cells might be too mesenchymal, too far gone to be efficient metastasizers, that you might need cells earlier on the EM continuum to metastasize efficiently. We first looked at the luminal model, either with a non-targeting or either of two hairpins against Vimentin, and what we see is consistent with its very low expression, its knockdown has no phenotypic consequence. You know, at image level or at quantitation level, there's no impact. We saw something very different in the basal triple-negative gem. On the left is your non-targeted shRNA, right? You see what's the baseline level of invasion, knockdown Vimentin. Again, this is an intermediate filament protein. At no point in my career did I say, oh gosh, I want to be the intermediate filament guy, but we keep getting led to them, we keep knocking them down, we keep getting really interesting phenotypes, right? You take away Vimentin, these cells cannot invade. We generalize that across two triple-negative breast cancer PDXs, same result, take away Vimentin, with either of two hairpins, they don't invade. We next wanted to test whether it was important for metastasis formation in vivo. You'll see in one of the next sections why we felt it was very important to independently test invasion and metastasis. But here, what we're going to do is we're going to take the tumor, isolate the epithelium, digest to organoids, either knock down a non-target, add a non-targeting shRNA or a Vimentin targeted shRNA. Allow the tumors to form and look at the lungs. And what we see is essentially complete abrogation of the ability of these tumors to form metastases. We saw one tumor in one mouse, and that still could have been shRNA escape. So we essentially completely eliminated their ability to form into bulky tumors. Somewhat more surprising to us, we also reduced their seeding of the lungs. Because you could have imagined that you knock down Vimentin and they get to the lungs just fine, but they don't seed. So instead what we see is they don't seem to be arriving, they don't seem to be expanding. So that was good. We then wanted to broaden our molecular lens from thinking exclusively about Vimentin, E-cadherin, a handful of protein markers, to thinking at single cell level about transcriptomes. And so what we did is we used, the way we thought about this was we're taking cells out of a primary tumor, and on average, any given organoid is not invading at the moment you harvest the tumor, right? Because it's a relatively small fraction of the volume that's actively invading at a given time. Chop and digest, drop them into collagen, a very high fraction of the organoids will invade. So the way we were thinking about that was we were inducing the invasive behavior. That probably meant that we were inducing the construction of the equipment required for invasion, and that this should be detectable at single cell level in the transcriptome. So we're looking at day 0 or 12 hours, day 3, day 5. We used 4T1 cells just as batch controls and did this through a series of mice. The first thing which jumped wasn't necessarily what we wished for, but that jumped was that among the patterns that machine learning revealed in the machine learning revealed in the single-cell transcriptomes was 3 different patterns that all strongly related to EMT. Yeah, no one in the rooms can be able to read the genes at the bottom there, so just look at the ones at the top. We have things like grainy head-like 2 in the first pattern, ZEB1 and ZEB2 here, oval-1 in the third pattern, but there's additional both effectors and transcription factors in each of these patterns, which we all think of as being pro-EMT. So we're seeing 3 different flavors of EMT. They're expanding to different degrees in invasion relative to colony formation, right? And we wanted to get an understanding of how these worked. It would be interesting to knock down every single one of these genes. We focused on the ones at the top. So we decided to test the requirement for ZEB1, ZEB2, FOXC2, oval1, and grainy head-like 2. The first 3 are pro-mesenchymal transcription factors. Oval1 and grainy head-like 2 are thought to promote the epithelial state. So with tissue from the same tumor, we're able to knock down each of these, so non-targeting versus each of these genes in both the colony formation and the invasion assays, so that we can get a sense of how loss of each of these transcription factors affects early relative to late steps in metastasis. So let me walk you through this one step at a time. Let's start on the lower left. Non-targeting shRNA shows you the baseline level of invasion in the assay, right? So you're seeing significant number of vimentin-positive cells, significant amount of invasion. Take away grainy head-like 2, we see a large increase in invasion, large increase in vimentin, right? Consistent with the idea that grainy head-like 2 is defending the epithelial phenotype. You take it away, system goes more mesenchymal, goes more invasive. Take away ZEB2 or FOXC2, large reduction in vimentin, large reduction in invasive behavior, consistent with the idea that both of these transcription factors are positively regulating vimentin and invasion. I want to highlight that ZEB1 and oval1 had no effect in this assay. Now let's look at later stages, right? And what we see is that ZEB1 and oval1, no effect on early stages, right, on the invasive behavior. Take them away, you get more colonies. So they're suppressing colony formation. Grainy head-like 2, ZEB2, and FOXC2, no effect. So what are we seeing here? First of all, don't assume just because the names are almost the same, that ZEB1 and are closely related evolutionarily, that ZEB1 and ZEB2 are doing the same thing in your system, because we're seeing really different utilization of very similar transcription factors in different processes of metastasis. More broadly, we're seeing that hybrid EMT cell states are driving basal triple-negative metastasis in GEMs, in patient-derived xenografts, and in patient tissue, and that the cancer cells are co-expressing both epithelial and mesenchymal markers, including some what are considered relatively early epithelial markers, echidherin, EpCAM, and some relatively late definitive mesenchymal markers like smooth muscle actin and vimentin. We showed that vimentin is required for invasion and metastasis, and that different transcription factors are regulating distinct EMT states during different stages of metastasis. So we think it's going to be really important. That's why I emphasize the decathlon model. It's going to be really important to be assaying what your gene is doing in different processes of metastasis, because it may help one and hurt another. The next thing we wanted to think about was heterogeneity in these tumors, right? Because we look at in this case, a basal triple-negative gem, and I can kind of find whatever I want to find here, which is distressing, and why we rely so heavily on both imaging and reporting these whole slide scans. Because if we look in this region, highly green, you're seeing that luminal-like pattern of very epithelial cancer cells and of vimentin-positive stroma. You go to one of the brighter red regions, and you're seeing totally different cell shapes, right? And you're seeing these ECAD vimentin double-positive hybrid EM cells. So that got us a little worried, because in data that I'm not showing, we knew that these different parts of these same tumors were invading with different patterns in culture, some more bulky, K14 positive, some more spindly and vimentin positive, and that got us worried that there could be different phenotypes in the metastases. I didn't want this, right? It would have been really nice if all the metastases did the same thing, and preferably if they all transitioned back to an epithelial state as predicted, because triple-negative breast cancer up to this point, just to be clear, standing in Boston, looked exactly like Bob Weinberg predicted for an EMT model, right? You have an epithelial origin, you acquire a bunch of mesenchymal gene expression, you spread through the body. The next step is that they should revert back to an epithelial phenotype pretty uniformly in the distant site. So what we did is we took the triple-negative breast cancer gem, we harvested the tumor, got it down to clusters, put it in the tail vein, and let's just look at the lungs and see what happens. Again, we were hoping they would all go back epithelial. We were hoping they'd be very similar. That is not what we got from either GEM, which I'm showing you, or human patient triple-negative breast cancer metastases. Instead, we see diversity of EM cell states. So what we're looking at here, look at the TD tomato is the labeled cancer cells that you can see distinct from the unlabeled background. Vimentin, we're looking for its absence because it turns out to stain the lung parenchyma. We tried a bunch of antibodies. We couldn't find one that was negative in the lungs. So you're looking for absence of vimentin. E-cadherin stains brighter in the metastasis than it does in the lungs, so we have better contrast there. So first row here, the metastasis is vimentin negative, E-cadherin positive. We'd call that an epithelial metastasis. Next row, the metastasis is vimentin positive, E-cadherin positive. You call that hybrid. Then we've got vimentin positive, E-cadherin negative, at least more strongly mesenchymal. And the worst part, the part I really don't like, gives me nightmares a bit, is these mixed states where within the same metastasis there's what looks like, and we think we're close to showing that it is, a clonal boundary, right, where different cells are expanding, which are holding on to different cell states in different regions of the same met. We see these mixed states in human triple-negative breast cancer metastases as well. So down here you have a hybrid ECAD vimentin double positive, as well as an ECAD negative vimentin positive region. So that's a little bit terrifying. Because what are we seeing here? We're seeing different cell states simultaneously metastasizing from the same tumor to form diverse metastases. I feel it's important to then say, we're very interested in understanding not just how could it be different, but how could there be common vulnerabilities, right? Can we find any common molecular requirements in these different metastatic strategies, right? Because if a patient can have multiple clones metastasizing by different molecular mechanisms, you don't want to have to target each of those individually. In an ideal world, you'd want to be able to find a common vulnerability of both strategies. So how did we get into that? Again, in the Wayback Machine, as soon as we found these keratin-14 positive cells, we decided it'd be really interesting to transcriptomically profile them relative to keratin-14 negative cells from the same tumor, right? We anticipated, as I think most people would have, that the keratin-14 positive metastatic pioneers would be downregulating most aspects of their epithelial phenotype and their epithelial cell adhesion. That was not what we observed. Instead, the keratin-14 positive cells spreading through the body are upregulating multiple distinct aspects of their epithelial cell adhesion machinery. So 5 to 6 log 2 on almost all aspects of the desmosome, 5 to 6 log 2 over almost all aspects of the hemidesmosome, in addition to retention of E-cadherin and co-expression of P-cadherin. So these cells are putting a lot of energy into making cell-cell and cell matrix adhesion complexes. They were making too many of them though, so I didn't know where to start except to start with E-cadherin. And the challenge we had there was two distinct literatures, one of which was coming from molecular biology and analysis of cell lines in culture, which had as the conventional model that E-cadherin suppresses invasion and metastasis. A reminder you probably don't need, intro genetics, if you say something's an invasion suppressor, you say that because when you knock it out or knock it down, you get more invasion. Knock it out or knock it down, you get more metastasis, would allow you to conclude it was a metastasis suppressor. However, most of the data on this was natural experiments where people took a cancer cell line and they looked, this is a two-sided bar graph, say, okay, this is a highly invasive cancer cell line. Look, it expresses minimal E-cadherin. This is a high E-cadherin expressing cell line, minimal invasion. And this held true, epithelial cancer after epithelial cancer. Their invasive behavior in culture was clearly anti-correlated with their level of expression of E-cadherin. And there was some functional work as well. So MDA-MB231 cells, very spindly and solitary, classical cadherin negative, force expression of E-cadherin, they form clusters, right? So that's, at the time I too thought that was less dangerous. Now with the work from the Haber lab, the Stanger lab, and our lab on cluster metastasis, it's at least unclear. But there was another literature running around, which is the pathologists who again and again kept finding E-cadherin positive tumors and even E-cadherin positive metastases. So invasive ductal carcinoma, one of the one of the several primary ways to classify breast cancer is into ductal versus lobular phenotype. And this is chiefly done by expression of E-cadherin, right, and the associated catenins. So you have invasive ductal carcinoma, about 90% of breast cancer, typically E-cadherin positive in the primary tumor. It's the brown stain here. You look at the metastases, they're retaining E-cadherin. Alan Wells at Pittsburgh would argue they're enriching for E-cadherin. It's a hard comparison to do between tissues, but they're certainly not always losing it. And it's hard to be a metastasis suppressor if you're overexpressing the gene in the metastasis. So we had this, honestly, itchy brain moment for me, lasted several years, of molecular biology is arguing it's suppressing metastasis, pathology is arguing it's still expressed there. So we decided to use genetics to sort this out. We used Crelock's genetics, Rolf Kemmler's floxed E-cadherin allele, Laquan Lowe's Cree biosensor. So we can cleanly delete the locus. It's an extreme experiment, but it was very informative. And I have to emphasize we're doing this in invasive ductal carcinoma models that would normally express E-cadherin in both the primary and the mets. So the first experiment is just to test whether loss of E-cadherin promotes invasion in IDC models, right? So we'll have E-cadherin intact, we'll take away E-cadherin, everything else is the same media image at the same time. Dozens of papers argued that E-cadherin was an invasion suppressor. You take it away, you should get more invasion. Dozens of papers were right. You take it away, there's clearly more invasion. If you look carefully at the image on the right, though, you'll notice it's not falling apart to single cells. It's not even only disseminating as single cells. There's large groups of cells disseminating. I'm also going to encourage you not to underestimate this guy just because it's a relatively modest invasive pattern in culture. In vivo, that will met to the lungs 95% of the time on a FVB background. And I should say, we tested this in both luminal and triple-negative breast cancer IDC gems, the same two that we've been echoing back and forth throughout. So this is a common dependency here. In both cases, the E-cadherin is suppressing invasion. We also tested in triple-negative PDXs. But that doesn't answer the question we were most interested in, right? We still need to test whether it's suppressing metastasis. For that, we need an in vivo system. So we create primary tumors. We're first going to look at invasion. Create primary tumors which either retain or lose E-cadherin. What you see is the ones that retain E-cadherin, they are suppressed in their invasion. It's only 6% of the tumor stroma border has an obviously infiltrating phenotype. You take away E-cadherin, it goes to 82%. You get these long, spindly chains of cells going out into the extracellular matrix, right, going out into the adjacent stroma. So large increase in invasion in vivo. Two different IDC models. But the question we were really interested in was whether it increased metastasis. For that, we take E-cadherin intact, E-cadherin deleted, let them form primary tumors, and look at the lungs. And what we found was that E-cadherin negative IDC cells do not efficiently form metastases in orthotopic or tail vein assays in either luminal or basal gems modeling invasive ductal carcinoma. We saw one met in one mouse. So we observed this uncoupling of the efficiency of invasion and metastasis in ECAD-deleted IDC cells. So more invasion, less metastasis. We had 4 vigorous reviewers, and so we were able to test a lot of intermediate steps in the metastatic cascade. Oh, you get vigorous reviewers here too, that's good. So they're worse at directional migration, they're worse at getting into the blood, they're worse at surviving in the blood, they're worse at getting out of the blood into the distant site, and then even if you normalize for that, right? So you have, they're separately, statistically significantly worse at both survival and proliferation in the distant site. I want you to think of this like the decathlon, where you win the 100-meter dash by 3 seconds and tear both your hamstrings and withdraw from the next 9 events, right? You lose the decathlon. So these are great invaders, they're terrible at metastasis. Why, right? So differential gene expression analysis, take away E-cadherin, see what's different. There's 129 genes there, a lot of them are interesting. 29 of them are involved in apoptosis, so we went like a laser beam on that. It turns out even in the bulk, green here is E-cadherin negative, red is E-cadherin positive. Even in the bulk, there's a statistically significant, fairly impressive increase in apoptosis in the ECAD negative cells, but it's when they disseminate. They start to leave, you'll see the blebbing, nuclear fragmentation pop. For those who don't like morphology, we did cleave caspase 3 positivity as well. Again, this is really pretty significant in the bulk. The tumor curve turns over immediately if you delete in vivo, but it's more than 50% are showing cleave caspase 3 positivity of the disseminated cells, right? So what's going on there? We tried 4 mechanisms that did not turn out to be the case until we got to looking at SMAD2/3 signaling, right? So we looked at where was SMAD2/3 when ECAD's intact versus deleted. We see, bang, it goes to the nucleus in the bulk and in the disseminated cells. TGF-beta 1 receptor 1 inhibitor rescues cell death. But that can't be the thing that changes when they disseminate. And so that turned out to be reactive oxygen. I can tell you in unpublished data, mitochondrial reactive oxygen is what's specifically being upregulated, and what, when inhibited, specifically rescues. So if we look here, E-cadherin intact, there's a little bit of enrichment, right? It's again, if you weren't looking at the slide, at the images below, it would look pretty cool. You're seeing an enrichment in cell and reactive oxygen in the cells as they disseminate, but it's stunning when you delete E-cadherin, right? The bulk's still pretty intact, but at the tips of the invasion strands, it's starting to increase. The cells that are disseminating, it's 10, 20, 30-fold increase in reactive oxygen. So how do we think this works? We think E-cadherin is functioning as an invasion suppressor, survival factor, and metastasis promoter, and that it's working as a survival factor by keeping TGF-beta signaling in check, preventing lethal accumulation of reactive oxygen or induction of apoptosis. And I want to highlight that this is a cell adhesion molecule, or at least we think of it chiefly as a cell adhesion molecule, that's regulating a pro-metastatic cell state. All right, so we've talked about a little bit of tumor microenvironment at the beginning, a bunch of cell autonomous mechanisms. Now let's flip back to the tumor microenvironment, but thinking about the cellular aspects of the tumor microenvironment. So the Keratin-14 positive cell, in addition to surviving in a cluster form as it travels through the body, also needs to evade the immune system, right? It's going to be bouncing into immune cells in the stroma. It's going to be surrounded by immune cells in the blood, bouncing into immune cells in the distant organ. So one of the things that completely surprised us and took us a couple of years to even think about how to think about was that the Keratin-14 positive cells are strongly downregulating at the RNA level the major histocompatibility complex, right, relative to Keratin-14 negative cells. And in fact, Keratin-14 on the x-axis, MHC class 1 on the y-axis in a FACS plot, they're mutually exclusive. There's almost no one in quadrant 2. So if you're expressing K14, you're MHC class 1 negative. The cheat sheet version of that, because I'm still a developing immunologist, is that cells without MHC class 1 are invisible to T cells and will be targeted by natural killer cells. So I got a terrific medical oncology fellow in the lab, and he said, I'm convinced NK cells are regulating this. Let's make co-culture assays. I said, we've never done a co-culture assay. It might not work. He said, it's going to be great. And it was. Overconfidence sometimes really is helpful, as long as your advisor can redirect you to a new project if needed. But it worked. It worked. So what did he say? So we got the spleen. We started off real simple. Spleens, you can isolate NK cells from the spleen. You can differentiate them. And you can then, from the same tumor, again, we get 2.5 million clusters, 250,000 organoids, 2.5 million clusters per tumor. So we can set up lots of experiments with lots of variables. So we digest into clusters, take a look at how do the clusters form colonies as monoculture versus how do the clusters form colonies when they're in the presence of NK cells. Initially, we're calling them healthy NK cells from spleens of just wild-type mice. So what do we see? Control has a certain baseline of colony formation. Add NK cells, you get a large reduction in colony formation. That's reasonable, right? That's what NK cells are supposed to do. And they're early responders to the cells that arrive in distant organs. Consistent with this reduction, you see a strong increase in apoptotic activity in the co-cultures. So it's not just that they're not forming colonies, they're dying, consistent with the idea of NK-mediated cytotoxic cell death. Even more consistent with that, if we take the tumors, fax them to K14 positive versus K14 negative, combine them with healthy NK cells for 4 hours, and just look at where is the interferon gamma expression being induced. It's in the co-cultures of the K14 positive, not the K14 negative, which is, I think, a pretty clean molecular argument that this system is working the way we would predict. The cells that lack MHC class 1, an inhibitory receptor for the NK cells, are the ones that are aggravating the NK cells, inducing them to kill. But here's the thing. Breast cancer patients, I think almost all of them, started off with reasonably efficient natural killer cells. By definition, if they're getting metastases, they must be breaking through this level of control. How could that work? And the way we got at that, simple experiment turned out to be very informative. We did this 2 ways. Either harvest the spleens, get the NK cells out of there, or, which is technically much harder, so we did it less frequently, but we got the same result, harvest the NK cell, the tumor-infiltrating NK cells, and put them in the co-culture. Same result, call both tumor-educated NK cells. So we're going to look at monoculture of the clusters versus co-culture of the clusters with tumor-educated NK cells. What do we see? Baseline level of colony formation in the monoculture, large increase, about twofold increase in colony formation when we add the tumor-educated NK cells. When I was a grad student, most cells of the immune system were already well known to have a pro and anti-tumor function. As far as I know, our paper here was the first to show a pro-metastatic function or polarization of the NK cells. It would have been an expected result for macrophages or neutrophils. But NK cells, as far as we know, were the first. So we next wanted to test this in vivo. We didn't want to keep it an in vitro conclusion for long because it was quite surprising. So the modality here is we're going to tail vein inject cancer cells, wait 24 hours, and then mock inject or inject healthy NKs or tumor-educated NK cells. Wait a week, look at the lungs. What we see is baseline level of lung metastasis, large reduction if we add healthy NK cells. I should have said this is non-SCID gamma mice or NSG mice. Therefore, there's no functional adaptive immune system. There's no functional NK cells. So these are the only NK cells we add are the only functional ones in the system. So healthy NK cells gives a strong reduction in lung macrometastases. Tumor-educated NK cells, large increase. Very consistent with our in vitro result. We next wanted to understand how this works at the molecular level. So we took our spleen-derived healthy NK, spleen-derived tumor-educated NK, did RNA-seq. And we got this RNA-seq here. And I don't know how it works in Boston, but in Baltimore, usually we see dark red, light red, maybe white, then light red again. There's usually variation across the lines. So we were really struck when we looked at this that we're seeing basically straight line, red, blue, blue, red. It was a very consistent phenotype. It immediately provoked us to think that there could be an epigenetic component to this. And in data I won't show, DNA methyltransferases are definitely regulating this cell state transition. What we focused on, when I say we, I mean Hildur Knudstoder and Joel Bader, was using known receptor-ligand interactions to take this differential gene expression analysis into predicted receptor-ligand interactions, right? So we could say, okay, what are all the likely ways in which a tumor-educated NK cell could be talking differently to a K14 positive cell versus a K14 negative cell? We tried, in that paper, we tried TIGIT, which is a known NK inhibitory receptor, and KLRG1, which is, I think, still an emerging NK inhibitory receptor. That one jumped out of our data, it's the red line there, because its known, its best characterized inhibitory ligand is cadherin-1 or E-cadherin, which could give an additional reason why E-cadherin positive cells could be favored during metastasis. So then we wanted to test whether blocking antibodies against TIGIT or KLRG1 could inhibit colony formation. And we tested that in a pretty simple way. So we have the clusters, tumor-educated NK cells, and we put them in culture, and we take a look at what the antibodies do. So what do we have here? We have a monoculture. We add the gray and the blue are both going to be co-culture, so monoculture and white. Co-culture with the IgG, and you see a twofold increase. First panel here is PD-1. We didn't have a reason to be doing PD-1, except that it was 2019, and you basically had to do PD-1 in your experiments whether you liked it or not. Neither low nor high dose had any effect on the system. But again, we didn't expect that this was a PD-1, PD-L1 regulated system. We did expect that TIGIT and KLRG1 could. So again, monoculture, IgG, low dose, high dose TIGIT, clear reduction in metastatic colony formation from high dose, which is not that high, of either TIGIT or KLRG1. So what did we learn here? We learned that the immune system is corrupted to promote metastasis. The NK cells are early responders to cancer cells arriving in distant organs. They're selectively killing what we've defined as the most invasive breast cancer cells. Cancer cells can rapidly corrupt NK cells to promote metastasis. In culture, it takes about 72 hours, and you can see that state transition. And inhibition of TIGIT or KLRG1 limits metastatic colony formation. This is a potential concept for immunotherapy in breast cancer. The fellow, Isaac Chan, is now an assistant professor in hematology oncology at UT Southwestern. He's pursuing that concept, and I think more preclinical work is needed to understand how it fits in with the other immunotherapy concepts that are available. All right, the last vignette I want to talk to you about is Claudin-low breast cancer. This is one that really highlights the sort of a secret technique we use, which is we go talk for a cell biologist. I talk to physicians a lot. And so we knew there was this claudin-low subtype of breast cancer. We knew it was a very aggressive breast cancer subtype, lots of invasive behavior, significant EMT profile, lots of metastasis. And it was characterized, it was found actually by differential gene expression analysis by Chuck Peru, and it was named for its low expression of claudins 3, 4, and 7. It's most abundant in poorly differentiated ER-negative tumors, but it can occur in essentially any subtype, which has actually led to a bit of an argument in the literature whether this is a subtype of breast cancer or a phenotype which can be layered on top of any of the subtypes. To the best of our knowledge, what hadn't been tested was whether the low expression of claudin 3, 4, and 7 had anything to do with their aggressive phenotype. I asked Chuck, and he said, we never thought to test that. Claudin was just a name we gave them. As cell biologists, claudins jump as important receptors that constitute the tight junction. We thought it could be interesting. Certainly very doable. So first, simple experiment, right? If we knock down claudin 7, the hairpins work well. Do we get more invasion? Well, yes, we get a lot more invasion when we knock down claudin 7. This was a bit of a home run experiment. You do it, but you tell the postdoc first, man, this could fail. It could give you nothing, which was to force expression of claudin 7 in a highly invasive context in which the cells would not normally express claudin 7. And what we saw was it really was individually sufficient to block invasive behavior. We next tested in vivo whether claudin 7 knockdown would increase lung metastasis. This is in the MMTV new model, minimal metastatic behavior at baseline, knockdown claudin 7, we see a large increase in lung metastases. So very clearly, claudin 7 is functionally regulating the invasive and the metastatic phenotype of these cancer cells. We then did differential gene expression analysis, and what we saw at the pathway level was changes in the same pathways that define the claudin-low phenotype in the clinical samples. It is not one-to-one identical, which we wouldn't expect from a single gene knockdown in a mouse model, but it is clearly regulating the same classes of gene expression, the same pathways, as are observed to be dysregulated in claudin-low breast cancer. The other thing that jumped for us was that there was a set of genes related to smooth muscle actin contractility and to certain aspects of the cytoskeleton and ECM that were all being coordinately regulated. And so we were curious, you always want to figure out which of your RNA things are real. We knocked down we have control shRNA, we knock down claudin 7, much more invasion, and all of a sudden the cancer cells, which at least in a luminal cancer wouldn't normally do so, are highly expressing ACT-A2 or smooth muscle actin, right? So this was stunning in culture, and we wanted to then test that in PDXs, go a little bit more realistic. Okay, you can see that. That came out pretty good, actually. So you can see that there's claudin 7 localization in this tumor consistent with the idea that it's forming tight junctions. If you don't like that image, I would refer you to the Western blot where you see high level of expression of claudin 7 in the PDX-A line. PDX-B and C, you really don't see any expression on the images, and you see very minimal expression on the Western blot, right? It was known that claudin 7 could vary in tumors. What was interesting was to look at what happened to smooth muscle actin in these same models. So here you're seeing a claudin 7 positive epithelial cancer cell population which is surrounded by a smooth muscle actin positive stroma, presumably myofibroblasts. What you're seeing in the PDX-B and C is a claudin 7 negative tumor with smooth muscle actin being expressed in the cancer cells. We next looked at a series of fresh primary human tumors. We explanted them to culture, and we stained for both claudin 7 and smooth muscle actin. The one at the bottom's striking because there's no claudin 7 and lots of invasive behavior, but there's more than one way that could land. I love this image because if you look at the top of it, just at the nuclear level, it's a more organized, it's not really a nice epithelium, but it's a much more organized, tightly packed set of epithelial cells. There's clearly claudin 7 at the cell-cell boundaries, right? And so you have this less invasive, more claudin 7. The bottom component of the same organoid has essentially lost claudin 7 expression, highly invasive, and that's where the smooth muscle actin is expressed. So both with knockdown and with natural experiments in human tumors, we're able to see this anti-correlation between claudin 7 and SMA. So what are we doing there? We've concluded that claudins are restricting breast cancer invasion and metastasis. In effect, claudins are doing what people thought ecadherin was doing, right? They're suppressing invasion, they're suppressing metastasis. Losing them seems to have no fitness cost, right? So you just get better at metastasis. So you knock down claudin 7, you increase invasion in vitro, metastasis in vivo. Loss of claudin 7 is inducing transcriptional changes similar, not identical, but similar to the claudin low breast cancer subtype. And so our model is that the tight junction is restricting invasion and metastasis, and its loss is driving poor patient outcomes. We're currently knocking down every aspect of that smooth muscle actin module. And I just want to give you a hint where our lab's going. Last slide. We're trying to define the cell states at single cell level in state space, so single cell RNA-seq and related single cell methodologies, and then use machine learning to map those states to tissue. So we can understand if there's a continuum of EMT cell states in a triple-negative breast tumor, where are each of those cell states that we can see in a UMAP projection located relative to the key anatomic features of the tumor, like invasion into the stroma, intravasation, pioneering the colony, sitting next to the T cells, right? We're then very interested in epigenetic regulation of these cell states. So we have a large analysis ongoing, integrating high-C, ATAC-seq, ChIP-seq, and RNA-seq to get the chromatin regulation, the histone methylation, and the changes in gene expression that drive these changes in cell state. We're then refining both from promoter analysis of key genes like claudin 3, 4, and 7, and our epigenetic analysis. We're independently identifying candidate transcription factor regulators of these cell states. And we're, throughout this process, trying to keep a good eye on druggable enzymes and antibody-accessible cell surface targets across the stages of metastasis. We can figure out where to aim to try to make metastasis, to either prevent or treat metastasis. All right, I want to thank the people in the lab who do the work. I want to thank our funders. I want to thank you for your attention. I welcome any questions you might have. Joan, great to see you.
That was great. Beautiful synopsis of all your really nice work. I'm wondering, in the case where you have the luminal tumors that can turn on K14 and other beta routines, when they're emerging out of the cluster, do you have any idea do you know what's driving that change in state?
So I have an unsatisfying answer that I'm sure of, and then basically to be continued. Yeah, right. But P63, right, which is the least surprising thing I could say, right?
What turns on, what's driving P63?
I told you it was unsatisfying. So that's where we really hope the combined epigenetic and transcriptional analysis is going to let us move past that. I think we're actually going to get there first with the claudins, because there's a limited number of transcription factors that are going to bind to claudin 3, 4, and 7. None of them are typically mutated. They're typically transcriptionally co-regulated. We have a pretty clear idea of what are the most likely transcription factors there.
Mechanical, because when you see it, when you
Oh, sorry, you were asking a different question than I thought.
all those, including.
Oh, that's a different question, but I have a good answer to that one. So we initially thought, because the collagen versus matrigel, collagen invades, matrigel doesn't. You're like, oh, this is an integrin ECM, specific ECM ligand interaction that's turning on, I don't know, SARC, right? ILK, something like that. And so we put them in matrigel. They form, if anything, they induce a more complete set of keratin 14 positive cells on the outside. So then we put them into just laminin 1 or just collagen 4, same result. Then we put them into agarose. They induce a beautiful set of K14 positive cells on the edge. So what we think we're seeing is a very, very ancient wound healing response. They're detecting an epithelial-free edge. They're dedifferentiating in a very similar way as a skin lesion when it closes, which also scrambles its keratins, dedifferentiates, closes the wound, because the thing that a metazoan cannot tolerate is a compromised epithelium. So it dedifferentiates, closes the epithelium, then it gets political again, redifferentiates in distinct cell types. So we think it's something very ancient there. Additional questions? Yeah.
Kind of correlate to the beautiful matrigel experiment. So in the case that you see those like environment induce like invasion of your cancer cell, do you think that those like a stimulation is a one-way ticket, or it when they go to like different environment actually can reverse the EMT phenotype you are seeing here? For example, if you have the matrigel surrounded the invasion spot, did they reverse? And sort of linked to that is that if that's the case, could that indicate like additional clue that usually people are thinking about, like, oxygen, nutrient, those that can as a direction of the tumor invasion things? Could these be part of that as well?
Sure. Simplest part first, we tried putting these cultures in hypoxia chambers. We didn't see any gross differences. We haven't followed up on it. On the question of if you switch them, We actually did that. That's the 2012 PNAS paper. We did with human and with mouse tumors, human and mouse normal. Grow them in matrigel, switch them to matrigel. Grow them in matrigel, switch them to collagen, 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 collagen to matrigel. It's their current local 3DM environment which determines their behavior. And in some slightly weird experiments that didn't end up in the paper, you carry along some incomplete digestion means that the same organoid has two different environments on it. Left side of the organoid can invade like matrigel, right side of the organoid can invade like collagen. So current, local, switchable. The thing which I think we're more generally asking is could the, which we have a more exciting answer to, is could the tumor microenvironment be determining the cell state? And specifically on the epithelial mesenchymal. This is a little earlier. I mean, we're writing up the paper, but it's a little earlier, unpublished. In the triple-negative breast cancer model, when the clusters are still strongly preferred. It's like 100 to 1 in the luminal. It's only like 30 to 1 preference in the basal, but 30 to 1 still wins. That means almost all the time that's what will show up clinically. The clusters do better. Very surprisingly to us, because we saw these diverse metastases, we knew we were seeding with clusters, we got a great suggestion from Rick Thompson. Super tedious to do, super, super informative, which he said, that's too interesting. You have to go, like, you can't just show me 4 weeks, right? You got to go a bunch of time points in between. That's a lot of mice. It's a lot of work. Amazingly worth it, because specifically in the triple-negative model, the luminal model, they go to the lungs, they land, they grow. Boring, stays in place, monotonic growth, in culture models as well as in vivo. Triple-negative, they land as clusters. They're strongly preferred as clusters for the first 48 hours, and then they shatter to single cells, migrate all throughout the lungs, and then become diverse metastases. And so one of the things we're most interested in right now is the diverse EM nature of those metastases due to cell intrinsic differences, or is it ecological niches determining their differential ability? The other fun thing to note on that, I asked the postdoc whether when there's this go versus grow hypothesis, you're either migrating or you're proliferating. So I was like, oh, they're breaking up into single cells, they're migrating through the lungs. Are they quiescent? She stained for Ki-67, 90% were positive. So they're going and growing, which I think could explain, mouse model caveats notwithstanding, some of the paradoxical chemosensitivity of triple-negative breast cancer, where there's a window where some of the patients can be cured. It's not a high fraction, but can be cured by more or less any modern chemotherapy regimen, right? What we think we're seeing there is when they're scattering, extremely high Ki-67 fraction should be sensitive to nearly any cytotoxic chemotherapy, unless you miss the window, right? And they're, instead of running around as single cells, they've landed, they've started differentiating, and now you've got a gemisch of 10 different cell states, right? Some of them feeding off each other in these mixed metastases, and then nearly any chemo will kill some, but not all the cells. So we think that's why there could be a window for some patients.
Maybe one last quick question. How do you think about metastasis to different sites?
Ooh, I don't have a good answer to that. I think about it a lot, and I don't have a satisfying answer for organotrophism. So maybe one, we just don't know. We have no experimental traction on that, despite years of thinking.
So I think you mentioned that the TIGIT-1 antibody inhibited the metastatic colony formation in vitro. Do you know if the TIGIT antibodies affect metastasis in vivo?
That's in Isaac's lab, not in my lab, so I don't actually know the answer. I know that multiple that was the first paper we wrote. I'm sure that's common for some people here, where we had like 5 companies call us within a week of the paper coming out, because a bunch of people are running TIGIT antibodies in breast cancer patients. So I think the answer, the clinic may lap the preclinical on that one. But they were thinking about it as a T-cell therapy, not T-cell and NK cell. So that's why I think it was interesting for them to think about doing clinical correlates on both populations.
We're right up on the hour. Andy, thank you so much. This was fabulous.